Hi everyone. So I'm going to talk today a bit about my PhD project, and this is part of it. So I'm looking at the Sirius Passet in Greenland and looking at the taphonomy. So what I've been doing is concentrating on a few different aspects of the Sirius Passet preservation. So this is just a geological map here showing you where it is in North Greenland. And the sediment we have there, it's sort of black slates that we have that have been metamorphosed, and we have a lot of chlorotoid porphyroblasts, which indicates low-grade green schist metamorphism. And there's two distinct fascies there. One is really mud-rich and one is quite spotted with sort of lozenges present. And on the right here, just some examples of the type of preservation you get. These ones here are Bunelis, and they're the most abundant in the Sirius Passet. So I have looked quite a lot on Bunelis and looking at the taphonomy of that. And now what we've found is that Bunelis is preserved mainly as casts and moulds, which are filled with microbial mat material, which has been silicified. So this SEM image here is from a cross-section through a Bunelis mould, and you can see all these sort of structures here, kind of cellular structures, which have been preserved. And we have here a microbial filament, which was an SEM image from the surface of the specimen. Now down here we have one of the guts, which is what I'm going to be talking about today, and I've done a lot of analysis on the guts. We've done some CT scanning of them, but we don't actually have the data at the moment, so I'm going to talk about the SEM analysis that I've done. And the fossil that I've been concentrating on is this one here, Cambrian, which has been described by Budd in 2011, and these are his reconstructions here. It's quite a simple external morphology that it has, but what's really interesting about this arthropod is that this internal structure is always usually preserved, and we have a lot of really nice preservation of that, which is what you see here. So what I've done is I've done some high resolution photography to try and map out the morphology of it, and this is a reconstruction which was done by Budd. So this part here is the gut tract, which runs up the centre, and here we have the diverculae which run parallel along the body and along the axis. And I've done some SEM EDAX analysis to work out the composition. Now, this sort of orangey stuff that you see here is mostly phosphate, calcium phosphate, which has been preserved. And the reason for that, we think, is that in the gut you're going to have a lot of high organic content. So at the time of the animal's death, there's a lot of organic content there to be preserved, which sometimes will precipitate calcium phosphate. And you can see here from the spectral analysis, got some silica here, but also quite a lot of the calcium and the phosphate there. But there is no evidence of the sedimentary composition of the matrix, so you don't have the typical matrix like the chlorotoids and everything that you see within the rock. So that suggests that it wasn't sort of filter feeding from the sediment. And here I've sort of mapped out some of the areas of the specimen so you can see it a bit more clearly. And all this stuff here that I've put in blue, that is actually preserved muscle structure. So we've got fibres preserved, and these are actually preserved as silica, whereas you've got the calcium phosphate and the diverculae, which would sort of essentially are kind of like the bowels of the organism. Now, that's quite a complex digestive system for an arthropod in the Cambrian, which suggests that it was probably a predator, so it was sort of eating more complex food stuff. And it means it was able to sort of filter the food throughout more efficiently. And this is some examples from the SEM which I've done. Now this one here, I got a cross-section made through one of the samples and looked at it, and you can see that it is just full of calcium phosphate. That's all the really bright stuff there. And it also appears as nodules, and this is a really high resolution one at 4,500 times. And a lot of it appears quite spherical and quite nodular, and this is something that I need to look at further, because we also have muscle structure, which, as I said, is preserved in silica. Now, these are really close together, so I need to work out why are we getting preservation of silica in one part, but preservation of calcium phosphate in the other. So you can see here from the EDAX spectra that the carbon's just from the coating of the specimen, but the silica and oxygen is just full of it. Now, on the surface of these muscle fibres, you can see these tiny nodules which could possibly be sort of microbial in origin. And because we've found a lot of evidence of microbial activity in Brunellus and the preservation of the mould and the casts, I want to explore whether or not this is something that is happening within these specimens as well, because obviously microbial activity does affect the preservation of organisms as they die. And this here is a picture of the gut tract, which higher magnification zoomed in, you can see all the phosphates. So the phosphate does seem to be concentrated in the actual gut itself, whereas the muscle structures round about are silica. Now, one possibility is that the muscle itself would have been able to withstand the decay a bit longer, allowing for this silicification. Alternatively, it maybe was calcium phosphate and then has become silicified. So if we can work out the timing of these events, then we'll get a better understanding of this taphonomic pathway. And the main conclusions here are that the distribution of the phosphate in silica are tissue specific. So you are getting different preservations in different parts of the organism. So what I'm going to do is, once I've got the CT data to look at, I'll get a better sort of 3D reconstruction of the morphology of the gut and hopefully be able to get some more information about the morphology and how that relates to the preservation. And because large areas of the digestive system have been phosphatized, this is something that's been seen in other organisms, in other lagerstatin as well. So it is likely that the theory that because of the high organic content from, obviously, if the animal has died after eating, then it's going to have a high organic content, which could precipitate the calcium phosphate. But the lack of the sediment or microbial mat material within the gut suggests that it probably was carnivorous rather than a deposit feeder. So overall, the reason I've looked at this one is because we have so many specimens, but we do have a lot of guts preserved from other arthropods and from lobopods and things as well. So what I'm going to do is do some comparative studies on them all, along with the CT scanning, so that we can try and get sort of a better timing of the taphonomy and work out exactly what was going on in the Sirius passet. And if there is evidence of microbial activity causing preservation in the muscles, it means that it might not be that distinct from the preservation of Brunellus, which is something that I've done a lot more work on, and we've just finished writing the paper on that, so that'll be submitted quite shortly. So it would be quite nice if the preservation of this did tie in and we sort of had a link between the two styles, because if you're getting some preserved as moulds and casts, then why are you getting some guts preserved and some not in Brunellus? So it'd be really interesting to find out what the link was between the two types of preservation. And this here is the hypothesis that we're sort of left with at the moment. And so I postulated that the precipitation of calcium phosphate in the guts is a result of the initially high organic content in the digestive tracts, and that the preserved muscle structure is mostly silicified, so it may be due to the sort of death masking, which was first described by Gehling, and it might be a result of microbial activity under localised conditions in the muscles. And any questions?